Hello everyone. In this video we're going to talk about the structure of DNA. As we know from the previous video, DNA is the hereditary material in humans and other organisms. First you need to know what DNA stands for. It stands for Deoxyribonucleic Acid. And this name is attributed to its structure, which you'll be familiar with by watching this video. So why is the name deoxyribonucleic acid? We're going to explain that later in the video. Let's start now with the structure of DNA. A DNA molecule consists of two strands coiled around each other, forming a double helix. This double helix, if we unwind it, is somewhat like a ladder. Each one of the two strands is a long polymer of simple units called nucleotides. So these are nucleotides. A nucleotide is composed of three components: a monosaccharide pentose sugar, which has 5 carbon atoms, 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. It also has a phosphate group and one of the possible 4 nitrogenous bases. So let's explain each part of a nucleotide. Let's begin with the nitrogenous base. A base is attached to the 1 prime carbon of the sugar. This base is the central information carrying part of the nucleotide structure. A base can be adenine, guanine, thymine, or cytosine. Adenine and guanine are called purines. While thymine and cytosine are called pyrimidines. And that's because adenine and guanine have 2 heterocyclic rings fused in their structure, while cytosine and thymine have just 1 heterocyclic ring in their structures. The typical arrangement of the bases is that adenine binds with thymine via a double hydrogen bond, and cytosine with guanine via a triple hydrogen bond. The second part of a nucleotide is the pentose sugar. It is called deoxyribose because it misses a hydroxyl group in the 2' carbon. Hence the name literally indicates lack of oxygen. On the other hand, this hydroxyl group is present in ribose sugar, which is the sugar of RNA. And this gives their names DNA deoxyribonucleic acid and RNA ribonucleic acid. Now we know why it is deoxyribo, but why nucleic? That's because it is found in the nucleus, as we know from the previous video. But we haven't explained yet why is it an acid. Just give me 1 minute and I'll explain that to you. But for now let's get back and continue our small talk about the other 2 parts of a nucleotide, which are the sugar and the phosphate group. The sugar, with its exposed oxygen, can bond with the phosphate group of the next molecule. They then form a bond, which becomes the sugar-phosphate backbone. This structure adds rigidity to the structure as the covalent bonds they form are much stronger than the hydrogen bonds between 2 strands or between the 2 nitrogenous bases. So nucleotides are linked to one another in a chain by chemical bonds between the sugar base of one nucleotide and the phosphate group of the other. The phosphate group attached to the 5' carbon of the sugar on one nucleotide forms the ester bond with the free hydroxyl on the 3' carbon of the next nucleotide. These strong bonds are called phosphodiester bonds. And the sugar-phosphate backbone is described as extending or growing in the 5' to 3' direction when the molecule is synthesized. There is also one other important term you should be familiar with, which is nucleosides. A nucleoside is a nucleotide but without the phosphate group. If purine and pyrimidine bases in cells are linked to a carbohydrate or a sugar, this forms a nucleoside that doesn't have a phosphate group, while a nucleotide has the three components we already talked about: the sugar, a base, and a phosphate group. So nucleotides, because they have a phosphate group, we call them the phosphorylated form of nucleosides. Now the time has come to know why DNA is an acid. The acidity of DNA is derived from the phosphate groups utilized in the formation of the molecules. There is an easily lost proton from the phosphodiester bond that contributes to the acidic nature of nucleic acids. So let's go back to the structure of DNA. We can see that the nitrogenous bases lie on the inside of the ladder, forming the rungs. And by pairing with each other, they form base pairs, having hydrogen bonds between them. While the sugar-phosphate backbone is on the outside of the ladder, forming the vertical side pieces. And this backbone is strengthened by phosphodiester bonds, which, as we said, are way stronger than these hydrogen bonds inside. Now we have two strands. This one is growing in the 5' to 3' direction. We call it the parallel. Or the template strand. While the other strand is growing in the 3' to 5' direction, which is antiparallel or complementary to the other. That's it for this video. Thanks for watching. If you liked the video, please hit the like button, and if you would like to watch more videos, hit the subscribe button. And if you have any questions, drop them in the comments below and I'll answer them all. Goodbye and see you in the next video.